Hi, everyone. Welcome to Ion Research 2024. This is our second annual Ion Research, and we are so thankful to everyone for joining us. So just a special thanks to our sponsors. We are sponsored by Castle Biosciences, as well as IDEA Biosciences, Aura Biosciences, TriSalus Life Sciences, Immunocore, Delcath, and Replimune. So thank you to our pharmaceutical. Dr. Koya, thank you so much for joining us today. We're so glad to have you and so excited to hear about some of the primary research updates for primary ocular melanoma. So thanks for joining us.
Well, thank you so much for this opportunity. It's really great to interact with this group, so many patients, family members, and loved ones that are really interested and involved, not only in the care of patients, but also in the advancements of this area. So that brings me great joy to be part of. So I'm going to go ahead and I just prepared a couple slides just to kind of people can follow what I'm talking about and not be a little bit lost in all of this information. I am an investigator for Castle, IDEA, and Aura Biosciences. What I'm here to do today is to talk about general ocular melanoma updates and touch a little bit about potential new developments in neoadjuvant treatments. And finally, update everybody on the radiation retinopathy trial that's called the Protocol AL, which is sponsored by what we call the DRCR-NET, which is the Diabetic Retinopathy Network. And they started out looking at diabetes, and now they have verged into a lot of blood vessel diseases of the retina, and now including radiation retinopathy. So before I get started, I just want to share some wonderful news is that finally our paper, I'm sharing the kind of the validation of the 15 gene expression profile and PRAME as an integrated prognostic test for uveal melanoma was just published day before yesterday. So you can see here, we're all very excited. This is the first paper by the CUG2 group. And I'm sure when you all browse, you will probably see your ocular oncologist involved there. It was a big effort on all of us to get this information. We enrolled 1,500 patients, and it really provides now a good framework and a good prospectively validated framework in order to kind of come up with studies going forward, enrollment for adjuvant treatments, etc. So what are the current clinical trials for primary posterior uveal melanoma? Well, first of all, the IDE 196, the IDEA trial for localized uveal melanoma, the Aura 011 trial for the intermediate melanocytic lesions or small choroidal melanomas. There's a talk about a neoadjuvant tebentafusp for uveal melanoma that is not yet recruiting. And then there's a new player, which is the sitravatinib. It's the Citra, which can be a potential future target. Right now, they're using that as a target for other cancers like lung cancer. They didn't have such a good result. But this medication, probably associated with another medication, may be a good potential future target. I'm sure they will look at it first for metastatic disease, and I know that we'll have Dr. Carvajal and his team talking about it a little bit later, but definitely something to be on the lookout. And then finally, I'm going to talk a little bit about that Protocol AL, which is the intravitreal faricimab or the fluocinolone intravitreal versus observation to prevent vision loss after radiation treatment. So I know the IDEA team is going to be talking about their product and what their findings are so far, but I just want to kind of bring to all of you the potential impact of this medication and this treatment if it proves to be successful as it's promising to. Number one is that possibly will improve globe salvage tremendously, and as a result, limit vision loss of patients associated with radiation treatment. And that will have a huge impact in quality of life, and especially for survivors going forward. It could change the systemic outcomes of these patients because targeting GNA11, which we know it is an upstream mutation, it could really improve the rates or decrease the rate of metastatic disease. So it would work both ways. It would be a primary treatment, and it would be also a neoadjuvant treatment in some ways. And its downstream effect could potentially improve patient survival, as I mentioned. The other one that we're excited about is the neoadjuvant tebentafusp. We know that tebentafusp or Kimtrac has been used currently for metastatic disease, but the company is now trying to think about it as a primary neoadjuvant treatment. And, and this is something that will probably be launched here in the near future. These are the enrollment criteria. As you see, they're going to start with very large tumors that potentially could not be treated other than by enucleation. And they will obviously make sure the patients are all HLA-A0201 positive, that we know only 50%, give it or take, of the population are positive. And they want to enroll, as they are validating this potential trial, all the patients that are in good health conditions. So that's one thing I tell all of my patients. I always say, stay healthy. That's what you can do to improve your odds in whatever we do going forward. And also, they want to make sure this person will be able to adhere to the treatment, etc. And as you all recall, this is the data for the tebentafusp in the metastatic uveal melanoma setting. And we just think that this could have another huge impact in possibly improving globe salvage, possibly limiting the vision loss associated with the radiation treatment. And the downstream effect could potentially improve patient survival. And then the question is, will there be selection bias by only using tumor size instead of genomic profile? And this has been something that, this company has been very honed into other things that are not the genomic profile. Now that we've had the GEP plus PRAME validated, it would be kind of interesting to see how it plays out as far as the validation and its usage going forward. Then the Aura Biosciences trial. This is something that I know Dr. Hopkins will be talking about here pretty soon. I think the exciting thing is that this is a phase 3 randomized trial, and we will be seeing how these patients then behave compared to no treatment. I believe that the new suprachoroidal injection will definitely optimize the drug delivery, and I'm excited to see these results. And this is the phase 3 study schema. The only thing is that it's a very intense treatment. It really needs a buy-in from the patient, and understanding that you may not be randomized to get the treatment. So you may be randomized to get observation, but obviously you will be taken out of the group of observation if the tumor keeps on growing, and then you will get the radiation or whatever else treatment your physician deems appropriate. Just letting you know that, the inclusion criteria is more based on growth. And this is something that in the past, I have to confess that I was personally a little bit against just having that as a criteria. But the company is giving us now some freedom to do biopsy the patients before enrolling them. And I think that that will really make the data way more robust, having that information. Again, this just kind of shows that not all tumors are created equal and not all patients are equal. So we have to account for that. And I think that that will be really a game changer for those folks that have very small tumors in a location that radiation can be potentially damaging. So stay tuned, because this is going to be very exciting. So potential impact of this trial is going to be the early treatment of small posterior tumors showing progression over a short period of time, expected low risk for vision loss, and possibly mitigating or eliminating the radiation, but the possibility of radiation if the treatment fails or if the observation deems that, the tumor continues to grow. The question is, again, are we testing the efficacy of the treatment for small melanomas or large nevi? And I think that now that the company is allowing us to biopsy the patients before enrolling them, I think that will be really a game changer, and it's going to make that data a lot more robust. Finally, the Protocol AL by the DRCR-NET is going to be very impactful. They're going to test what we call the intravitreal faricimab, which is already commercially available as Vabysmo. This medication has been approved for age-related macular degeneration, as well as the fluocinolone acetonide implant, which is called the Iluvien, also by another company, but also already available commercially. So we know both drugs are safe. And then the third group would be the observation for the prevention of visual acuity loss. And so they want to see how does that impact the blood vessels around the radiated area? How does that impact the occurrence of blood vessel events, like clogging of the veins and the arteries as a result of the radiation? It's going to be a very interesting trial because right now we don't have any FDA approved medication for radiation retinopathy, and many of us do use Avastin, sometimes Eylea, but we have to jump through a lot of hoops to be able to approve it for the patient, especially if it's a younger patient that doesn't have any other diseases or comorbidities. So having that data will really push the FDA to approve certain medications directly for radiation retinopathy, and we're very excited about that. Here's just the things that we'll be doing, the standard visual acuity, slit lamp exam, et cetera, et cetera. And we will be monitoring patients every 3 months. The group that gets injections will get injections every 3 months, and the group with the implant will get 1 implant every 24 months. And the observation group obviously is not going to get any treatment. The downside of this study, I have to alert everybody, is that they are not going to be enrolling any patients with tumors around the macula, so around the focusing point and around the nerve. Still, it's going to be useful information. It's not going to be as impactful as we would like, but I think the idea behind the protocol would be some very striking, measurable outcomes. And that is the only way to avoid the bias of the tumor location. So that's the whole catch here. So the impact of this protocol will be providing insight into the benefit of preventative medication to avoid or minimize radiation retinopathy. And because of that, improving patient visual outcomes and hopefully quality of life, especially for those surviving the cancer, determine the success of the treatment for radiation retinopathy, and determine the frequency of treatment. That right now, we treat kind of by year, not really by year, but by experience and by feeling for how the patient is coming along. But we definitely need a protocol, something that can be followed and something that, as I said, can be used to get insurance approval. So I believe that that's basically my message. Over a century, we had no unchanged outcomes of patients with uveal melanoma, and we had limited sight-saving therapies. And now we're seeing a new era, and it's just so exciting. And I think these webinars are very, very helpful so everybody can stay tuned and understand what's coming down the pipeline, especially for the clinical studies. These clinical trials will likely change how we manage patients, and also will change how patients do after treatment in the long term. And this is the first step to improve survival of patients and improve quality of life and their visual outcome. Thank you so much.
We are so thrilled to have you here today, Dr. Cole. Thanks for being here.
Well, thank you very much, and I appreciate the opportunity to present what we're doing at IDEA Biosciences for uveal melanoma. So this is our safe harbor statement, just to let you know that everything that I'm about to discuss is investigational, and our molecule is not yet FDA approved for marketing. So I briefly just wanted to show everyone our pipeline, and the focus of my talk today will be this top part here, darovasertib. This is what we are developing for uveal melanoma, but we do have a robust pipeline of other molecules that we are developing for other malignancies. Very quickly, darovasertib. How does darovasertib work? As we know, most uveal melanoma is driven by mutations in these 2 genes over here, GNAQ and or GNA11. And these genes, with their mutations, propagate the signal through PKC, through RAF and through MEK, which produces our tumor, which produces our malignancy. Darovasertib stops that signaling or inhibits PKC right here, preventing that signaling from moving forward and also preventing growth of the tumor. So our first study in terms of primary uveal melanoma, we're looking at darovasertib in the neoadjuvant setting, darovasertib in the adjuvant setting, and a new protocol amendment for this trial is looking at the combination of darovasertib plus crizotinib in the adjuvant setting. So here's a schema of our trial, and I would say that the overall goal of our trial, we have a few of them, but we're divided up into sort of two cohorts, if you would. There's the enucleation cohort. The goal of that cohort to see if darovasertib can save the eye. The brachytherapy cohort, the idea being darovasertib, can it reduce the amount of brachytherapy that's given to the eye? And in the adjuvant setting, sort of afterwards, as we say, to save lives. So our primary endpoints in this phase 1/2 trial or phase 2 trial, eye preservation, decrease in radiation dose, and then secondary endpoints, useful vision, meaning improving the visual acuity or not loss of visual acuity. And I would also say relapse-free survival. So in our darovasertib company-sponsored trial, very, very preliminary results here I'm able to present to you. As everyone knows, there are a couple different ways in which we can measure decrease in size and tumor, whether it be apical height, basal diameter, and or the volume. Happy to say in our initial look at our data, when we look at 8 patients who've received greater than or equal to 4 months of treatment with single agent darovasertib, as we see over here, the majority of these patients, we have a median reduction when we look at this by volume of about 72%. And if we look at the light blue, these are our enucleation patients. If we look at the darker blue here, this is our plaque eligible patient. These patients have had major reduction in the size of their tumors. And we take a pause here for a second. The idea behind that is a patient who potentially was scheduled for enucleation now doesn't need enucleation. A patient who is scheduled for plaque brachytherapy now be getting a smaller amount of that plaque brachytherapy. And we'll have some examples of that moving forward. So if we take a look at a couple of case studies, a 50-year-old patient, uveal melanoma, receiving 4 months of darovasertib treatment. They have a dome-shaped tumor. If we look over here at the baseline, their baseline measurements, about 5 millimeters in apical height, almost 15 millimeters in the basal diameter, and their volume is shown below. When we took our measurements, pretty dramatic decrease in the size of their tumor, whether it be apical height, basal diameter, and or the volume, north of 40% decrease or upwards of greater than 80% decrease total. When we look at another patient who is scheduled for enucleation, the first patient was scheduled for plaque brachytherapy. Further on, we'll see some more slides about why we're excited about this. But here's a patient who is scheduled for enucleation. We see their initial baseline measurements over here, 8 millimeters, 13 millimeters, and almost 740 cubic millimeters of volume. After 4 months of treatment, we see pretty dramatic decrease in this tumor size, whether it be almost 3 millimeters in apical height, 8 millimeters in basal diameter, 90 cubic millimeters, showing percent change from baseline, almost 70% for the height, 40% for the basal diameter, and almost 90% for the volume. When we look at some data that has recently been presented at ASCO, and this is data from Anthony Joshua, this is an investigator-initiated trial or an IST, we see the same treatment, darovasertib, in the neoadjuvant setting. This is a spider plot over here. And again, Dr. Joshua is looking at volume 2 over time. We see with these 15 patients, all the patients, except for maybe one initially from when he took the sample size, when he took the data, all these patients are showing a decrease in the volume of their tumor sizes. When we look at tumor volume in terms of eyes preserved of 12 patients, 75% of those patients had their eyes preserved. Again, light blue being eyes saved, dark blue, the one patient that was enucleated. And we see the median reduction again in terms of tumor volume is about 47%. How does this translate for our plaque brachytherapy patients? Again, more of Dr. Joshua's data that was just recently presented at ASCO. Take a look over here. We have a patient. This over here represents our tumor. Critical structures here, optic nerve and fovea. We see this as our baseline measurement, and we see how close the tumor is to these critical structures. Again, after treatment with darovasertib, we see a very large increase in the distance of the tumor to those critical structures, whether it be the fovea and or the optic nerve. So we can see how much here the tumor is actually shrunk, greater distance. Why is this important? When we look at the potential radiation field for plaque brachytherapy treatment, baseline, we see how close that radiation field is coming to those critical structures, whereas after treatment, we see that radiation field is much smaller. Over here, we have some more specific numbers of that. So if we thought about this patient initially, the dose of the optic nerve and or the dose of the phobia, when we look at the amount of radiation therapy that was about to occur, 78 grays, 157 grays. Post baseline, this is now decreased to 13 grays. That's an 83% change. To the fovea, 59 grays, which is now a 62% change. Now, when we look at the probability of visual loss from 1 year and or 3 year through the Aziz criteria, we see that at baseline, there was almost a 70% chance of visual loss and or a 95% chance of visual loss at 3 years from the initial settings here. However, post baseline, after treatment with adrovisertib, that possibility of visual loss at 1 year is now down to 20%. 3-year potential loss is down to 43%. And this is where I will end this part of my presentation, because the next part is for what we are doing in metastatic melanoma.
We have an update from Dr. Hopkins from Aura Biosciences regarding their COMPASS trial. Dr. Hopkins, thanks so much for joining us.
Tremendous. Thank you so much, Danae. It's such a pleasure to be here and just see the incredible activity in research and care for patients with uveal melanoma. So thank you for the opportunity to be here. I'm Jill Hopkins. I'm the Chief Medical Officer at Aura Biosciences, and I'll walk you through our current program and our hopes for changing some of the paradigm of care for people living with uveal melanoma. So disclosures, much like Dr. Cole and IDEA, we are an investigational virus-like drug conjugate. We are in clinical trials and are not yet approved for use. So Bel-sar, or AU-011, but we call it Bel-sar, is a novel viral drug conjugate. And this is a very novel technology that uses a virus-like particle. So this is similar to vaccines using a papillomavirus, but this is a non-replicating or empty viral capsid, doesn't contain genetic material. And we're able to demonstrate that it has the potential to have multivalent binding to these heparin sulfate proteoglycans on solid tumor cells. And this is important because these HSPGs, as we call them, are present across a range of tumors. So somewhat agnostic to genetic subtype, solid tumors present these HSPGs on the tumor surface, and we're able to see these VLPs bind to them. We then add to that a light activatable molecule. We conjugate that on the surface of the VLP. And when we activate that with a laser, we're able to see selective binding of our drug to the tumor cell and additional selectivity brought about by the activation only being at the site of where the laser is administered. So we're able to see very on-target tumor effects with a much better looking side effect profile when we look at trying to treat early tumors. So we know that this allows for a dual mechanism of action within the Bel-sar activity. So we have one mechanism where as we bind that VDC to the HSPGs on the tumor cells, we see tumor cell necrosis after light activation. So the tumor releases these oxygen species, it disrupts the cell membrane, and we see tumor cell killing via mechanism 1. We secondarily see as mechanism 2 that these damaged tumor cells release neoantigens and these DAMPs, we call them, damage-associated molecular patterns, and these bring immune cell reaction into the tumor locale. So here we're able to see necrosis or cell killing at the time of the activation, and then the secondary mechanism whereby immune cells come in. And here we're very excited by the possibility that long-term adaptive immunity can help with ongoing tumor control. So what are the goals of treatment? Well, as Dr. Correa mentioned, we are in phase 3, which means we're in a stage of development that we are hopeful will lead to registration and ultimate access of this therapy for patients. And the hope with Bel-sar is that we'll be able to provide local tumor control, we'll preserve vision, we'll move away from the radiation-related morbidities that are well known to the community, and that we'll see potentially an opportunity to treat early and reduce the risk of metastases with a side effect profile that will hopefully improve quality of life for patients. So we're very excited by the potential here. We are looking at the delivery of Bel-sar by injection into the eye. So this is done in the office. Do not need to go to the operating room for this. We use a suprachoroidal injector on the white surface of the eye. We deliver the Belsar in that location, and we do 2 injections on either side of the tumor. The blue drug, as you see there on the slide, kind of distributes back around the back of the eye, surrounds the tumor and distributes within it. And we then do 2 laser treatments. They take about 5 minutes once the drug has had time to distribute in the tumor. Again, all done in office. As Dr. Crea mentioned, it's a lot of upfront activity. You do have 3 cycles of treatment at the start of the trial, and you're then followed for an additional 15 months. But in office, and as we say, the potential for a side effect profile that allows for preservation of vision. This slide talks about how we're currently managing uveal melanoma. And I think whenever I look at this, and what this webinar absolutely highlights, is the activity in research and advancement across the entire spectrum of care for uveal melanoma, which I think is the most exciting thing about this slide. I think where we are seeing the potential for Belsar is in this local or early stage of disease, when we have either these indeterminate lesions or the small melanomas, where many people are in that watch and wait. So many people listening or with family members impacted by this, you may have been told at some time that you have this pigmented lesion in your eye. We're going to watch and wait and see what it does over time, because available therapies are associated with challenging side effects. It's always a trade-off of sorts to say, do I treat now and know that I'm putting someone on a radiation-related series of complications, or do I observe and make sure I'm seeing growth, or determining that this is in fact going to be a lesion that will require treatment? And I think that's a very important point of what Dr. Crea mentioned in her talk. We do in the trial want to show evidence of growth, because that is a somewhat irrefutable sign of change in the tumor. So to make sure we are treating true melanomas rather than nevi, that growth aspect is important. And as she mentioned, biopsies prior to entering the trial are absolutely fine to determine additional information to help support that. But we are, the thought of being able to move into lesions that are earlier, smaller, that might be in that watch and wait aspect now, with a treatment being explored in phase 3, is very exciting to us at Aura, and we hope to the community as well. Dr. Cole did a lovely job describing where IDEA is in terms of the potential in larger tumors, and then the systemic therapy we'll hear about in the next session. I think as I mentioned, the high morbidity associated with the current standard of care, which is radiotherapy and has been the case for decades, and it's successful in stopping growth of the tumor. The unfortunate side effect for people living with that is that the vast majority of patients treated with radiation do go on to legal blindness over time in the treated eye. So we really would be excited to see a paradigm shift that says we can treat early, we don't have the vision loss associated, and we can look at a shift in how we've been managing patients for many years now. Tell you a little bit about our phase 2 trial. So the phase 2 trial is completed, and the data from this allowed us to move to our phase 3 registrational program. In phase 2, we again looked at suprachoroidal administration to the eye, activated with laser, and this was a dose escalation trial. So we were able to move quickly through cohorts to show that increasing either the dose of the Belsar or the number of lasers was not associated with any side effect profile that prevented increasing. And we were able to increase up to our therapeutic regimen, which is what we're exploring in phase 3, which is 3 cycles of treatment, 2 injections, 2 lasers. And our endpoints were looking at evidence of tumor progression. So did the tumors grow over time? Was there any loss of visual acuity, measured as 15 letters or 3 lines on the eye chart from baseline? And any change in the rate of growth of the tumor over the course of the treatment cycle? And what we saw was 3 key outcomes that allowed us to move into phase 3. First, 80% tumor control rate. So when we looked at our therapeutic regimen, so the 3 cycles that are being studied in phase 3, we had 80% of those patients had a complete cessation of tumor growth at the 12-month mark. Compared to our subtherapeutic, that's not a true sham in the sense that patients did receive some drug, but we know it was a very low level of drug and laser, where only 20% of those tumors achieved control. So we had an 80% tumor control rate. Very importantly, we were able to demonstrate that 90% of the patients in the phase 2 trial maintained their visual acuity. So the vision with which they entered the trial was the vision with which they left the trial. And very importantly, these were tumors, and 70% of them were tumors that were very close to the macula or the optic nerve, which Dr. Cole nicely outlined the importance of those structures in maintaining vision. So even tumors in that high-risk location for vision loss, patients were able to preserve vision with the Bel-sar treatment. And then we saw a very low rate of adverse events in the trial, again, supporting our hypothesis of the dual selectivity of this treatment. We had a little bit of mild front of the eye inflammation that was managed with topical drops over a few weeks and resolved without any additional sequelae or long-lasting side effects, and no other major side effects reported. So we were very encouraged by the favorable safety profile coupled with the tumor control rate and the preservation of visual acuity coming out of our phase 2 trial. So this has allowed us to move into the phase 3 COMPASS trial. We are looking at these small choroidal melanomas or indeterminate lesions, and we are randomizing patients 2 to 1 to 2, which means within the main comparator groups, we have the 80 micrograms, the high dose of Bel-sar, compared to the sham control arm. And that is an observation arm. This was very important in our conversations with the FDA and the European regulatory agencies, that we do have treatment compared to sham or observation, which is for many of these small lesions what the current standard of care is. And then we have the 40 microgram, the lower dose of Bel-sar in there for masking, so that patients and physicians are not aware of which dose they're receiving. This is very important, although it sounds complicated. It's very important in terms of a registrational trial that allows for regulatory approval and access for patients. And we're pleased to say we have both fast track and orphan drug designations. We have a protocol agreement with the FDA and support from the EMA on our endpoints. So we're looking at enrolling 100 participants around the world. We have sites in North America, Europe, Middle East, and Asia Pacific. So we are truly global in our approach here. 90 sites to enroll 100 patients, with the hopes that we will have regulatory approval following enrollment and a 15-month primary efficacy analysis. So ongoing. And this map shows, you can see both on the Accure Insight website. Thank you, Danae. There's a really nice link to the COMPASS trial and any information you want there. We're also on clinicaltrials.gov. And this is a map of the current sites that are open and enrolling in the United States. Still more to come there. We've just been approved in Canada, the United Kingdom, and Australia, and we're following in short order with European sites as well. So a lot of activity on a key trial, and happy to take any questions. Thank you again for the opportunity to share our goals at Aura to change how patients with melanoma are treated. Thank you.
Hey, Danae, how are you?
I'm great. How are you?
Good. Thank you for having me.
Of course. So, Dr. Carvajal, we are so excited to chat about metastatic uveal melanoma. I know this is a hot topic, but do you have any specific thoughts that you want to give us, kind of going from this last section into our next section?
Yeah, that's exactly actually what I want to focus on, Danae, because I think your point that, we're really in a new place in this ocular melanoma field. And I have to say, as I kind of am speaking to patients and other researchers and clinicians now, I am so much more optimistic. I was always optimistic, but the degree of optimism is just much higher. And it just reminds me, I think some of you who've heard me speak before, I kind of talk about sometimes where we were in skin melanoma when I started as an oncologist in 2000, where we really didn't have anything, and fast forward 20 years later, with our therapies, we're curing patients with metastatic melanoma, not everyone, but 40 to 50% of them, with these novel immunotherapies and these novel targeted therapies. And as you said, as these effective therapies became approved, we had more license in terms of, I would say, creativity in the forms of novel clinical trials, novel combinations, which really just accelerated our ability to improve outcomes. And I think that's where we are now for uveal melanoma. So I'm really excited.
Well, I'm so excited to hear what you guys have, both you and our future guests that we have coming on. So I'm going to go ahead and let you share your slides, and I will step away so that you can present and we will get going.
Perfect. So I think, so this talk is maybe a little bit different. So what I want to do is maybe talk a little bit about that bridge, I guess, between kind of treatment of the primary, treatment of metastatic disease. And this is more of a conceptual kind of overview, because again, I do think we're moving towards cure. The way we're thinking about this disease and developing therapies, it's much different than the way we were thinking about things 5 years ago. And so this will be a little bit of a different talk than I usually do. Here are my disclosures. I do a little bit of consulting, a little bit of work with IDEA and Aura. And Jill and George, thank you for your presentations. So let me start with this, and this is conceptual. If we look at cutaneous melanoma, we have been able to increase the number of patients that we can cure through kind of 3 large buckets, I would say. And the left bucket is that with our novel immunotherapies, targeted therapies, and so forth, we're actually able to treat patients with widely metastatic cutaneous melanoma. So this is skin melanoma that's gone to the liver, bone, lungs, wherever, brain. And we can treat them with these immunotherapies, and we can just eradicate it, right? We still have work to do. I want to be able to do that for 100% of people, but 20 years ago, we were curing maybe 5% of people with metastatic disease. Now it's up to 50%, okay? And so one path towards increasing the number of people we can cure with uveal melanoma is just develop more effective therapies that work in the metastatic setting. Okay, and so that's kind of that left bucket. The right bucket is using various tools to prevent the development of cancer in the first place, right? And so for skin melanoma, which we know is predominantly driven by UV radiation exposure from the sun, if we use sunscreen, if we use sun-safe measures, stay out of the sun during kind of noon, wear wide brim hats, all of that stuff, we can reduce the risk of developing disease in the first place. And frankly, that's what we have to do for all cancers, right? Prevent it. I would say, where we are for metastatic uveal melanoma, we have some approved effective therapies. We have to enhance what we're doing, right, to actually be able to achieve cure in a high number of patients. In terms of prevention, we have a bit of work to do because, to prevent cancer, it's helpful for us to know what causes it. And that's something that we still have work to do, okay? But for this talk, what I want to focus on is the idea of eliminating what we call micrometastatic disease, right? And so can we treat patients, can we treat people with primary uveal melanoma, not just with enucleation or radiation, but can we use the medicine either before treatment of the eye or after treatment of the eye to kill any cells that may have escaped and might be hiding out in the liver, right? And that's what we call micrometastatic disease. And we know in skin melanoma and a lot of other cancers, breast cancer, colon cancer, if we give medicine around the time of surgery, either before and or after surgery, we can dramatically decrease the risk of the cancer coming back, and in some diseases, help people live longer. We can do that in skin melanoma. And I think we're getting close to the point of being able to do that in uveal melanoma. Okay, so that was a big overview. So if we take that kind of middle bucket, how do we eliminate micrometastatic disease? There are basically 3 ways that we can think about developing that, right? And one way we can do that is take what we know works in people with metastatic disease and then try it either before treatment of the eye or after treatment of the eye or in combination and see if it can kill off those rogue cells that might be in the liver, reduce the risk of the cancer coming back in the future, and improve outcomes. And I'll talk a little bit about the tebentafusp story in that space. I'll go through 2 and 3 kind of a little bit later, but so let's focus on this. In the metastatic setting, there are kind of 2 systemic approaches that we think about. One is tebentafusp. You've heard a bit about this, and that's an immunotherapeutic approach. And the other is the darovasertib story, which is the targeted therapy approach. I'm not going to talk too much about the kind of mechanism of darovasertib. George did a fantastic job about that. I would just say that the targeted therapy approach is basically looking at the signaling pathways within the cancer cell, right? Like the things that are telling that cancer cell to grow and spread and do the things we don't want it to do. And with these pills like darovasertib, we can shut down those pathways. It kills the cancer cell, improves outcome, okay? With the immunotherapeutic approaches, what we're trying to do is somehow increase the ability of our immune system, our immune cells in the body to recognize and kill off the cancer. And I don't think we have an immunocore speaker, so I'll talk just briefly about the mechanism about this. It's kind of interesting. Tebentafusp is, in this picture, it's this blue thing here. And here we have the melanoma cell up top and an immune cell there here. And this blue thing, this TEBI thing, it's what we call this bispecific molecule, where one half of it binds to this protein, GP100, that's present on virtually all uveal melanoma cells in high concentration. And if it's expressed in a certain way, in the context of what we call HLA-A0201, TEBI can bind to that really tightly. So binds to the cancer cell really tightly. And then on the other end, it has this thing that just attracts immune cells that are in that environment. And basically all it does then is TEBI brings the immune cells just in close proximity to the cancer cell, and then the cancer cell dies, right? So that's pretty cool. And it's really cool that this was the first drug that was ever approved for the treatment of uveal melanoma. And this is just in January of 2022, right? Like, why am I so optimistic? There was nothing that was ever proven to meaningfully improve outcomes in this disease before that, which is mind-blowing to me, right? that was the case. But finally, we have, our first approved systemic agent that really meaningfully improves outcomes in our patients with advanced disease if they are HLA-A0201 positive. Okay. Now I want to kind of shift to how are we applying this in like that preventative type setting. And one of the things that we noticed when we treated patients with metastatic disease is that the likelihood of significantly shrinking the cancer wasn't that high. And so up top here, this kind of bar thing, this is what we call a waterfall plot. Every patient is a bar, and if the tumor shrinks, the bar goes down. If the tumor grows, it goes up. And you can see that we have major shrinkage in orange in a small fraction of patients. And we have more modest shrinkage, so a little bit of shrinkage, or maybe even a little bit of growth in the majority of the patients. And then we have major growth in kind of a small subset. In some cancers, drugs that shrink cancer better leads to improved outcomes in terms of having the people live longer, right? Which is ultimately what we want to do. But for TEBI, we didn't see that relationship really. So whether or not you shrunk tumor, the outcomes were kind of equally good. And so even if you had minor shrinkage or maybe even just a little bit of growth, there was still that sort of benefit in helping people live longer. And so one of the problems with TEBI then was, well, if size doesn't predict overall outcome, right? If our ability to shrink the cancer doesn't predict overall outcome, is there a better way for us to kind of assess that rather than scans, which is usually what we do? And so what we did in the earlier trials is we collected something called circulating tumor DNA. And circulating tumor DNA, basically the DNA is kind of like the brain of all cells, and all of our cells have DNA. And DNA can constantly shed from the cells, and you can detect it in the blood. If we do a blood test, we can find fragments of your normal DNA. But cancer also sheds DNA, and we can separate the cancer DNA from normal. And so what was kind of interesting here is that in all of the people we treated with TEBI, if the ctDNA decreased, or if they cleared the DNA, if it just disappeared and we couldn't find it anymore, all those patients did well. And so there was a really strong correlation between our ability to get rid of that circulating cancer DNA and improved outcomes. And so based off of that data, the first of the adjuvant TEBI trials, and adjuvant just means treatment with something after treatment of the eye. So the first of these kind of adjuvant trials was developed, and this is being led by Mark Middleton, Paul Nathan, and Joe Sacco in the UK. And what they're doing here is they're identifying individuals with uveal melanoma that's high risk by cytogenetics or gene expression, usually it's cytogenetics and large size, who are treated, who are HLA-A0201 positive, because this drug only works if you are O201 positive. And then they check for ctDNA over time. If they detect ctDNA, then those folks get 6 months of tebentafusp. And then they can see, if we do that, can we actually clear or reduce the amount of ctDNA under the hypothesis that if we decrease the ctDNA, overall outcomes will be better, likelihood of the cancer coming back will be lower. So this is kind of one kind of ongoing trial. It's ctDNA based. It's a big trial, but it's still going to be a little bit hypothesis generating. But what we wanted to do was really design a trial that would more definitively answer the question, if we do TEBI after treatment of the primary, can we improve outcomes? And so this is probably 7 or 8 years in the making, and it's finally going to open up this fall. But we are beginning this trial called the ADAM trial. It's an adjuvant TEBI trial where we're going to take individuals with high risk uveal melanoma, A0201 positive, without evidence of metastatic disease, and they're going to be randomized to either 6 months of TEBI or observation, and then we're going to follow. There's no placebo here. You know if you're getting the drug or if you're on observation. Observation is the standard of care, right? So that's why we feel like this is a reasonable trial to do. And so this will be sponsored by a group called the EURTC, so it's a European group. We're going to open this up in select sites in the US as well, hopefully up to 10 sites, at least 5, and hopefully those sites will be open hopefully no later than early next year. I also just want to highlight that we do have what we call regional therapies that can be very effective in this disease. So because recurrence tends to occur in the liver, and in some cases, in many cases, it's what we call liver predominant, the bulk of the cancer is in the liver. A lot of times what we do is think about, is there a treatment we can put right into the liver that can fight the cancer? And there are a lot of different ways to do that. There's one technique called intrahepatic, isolated hepatic perfusion, and there's another one called percutaneous hepatic perfusion. The concept between both of these techniques are kind of the same. Basically, we isolate the liver vasculature, right? So you kind of put the liver on like a bypass pump. You put in a lot of chemotherapy into the liver. And because of that bypass pump, the chemotherapy doesn't go into the rest of the body and cause a lot of side effects. And with that high dose chemotherapy, we can see some really nice tumor shrinkage. And so this PHP platform by Delcath is the second treatment that was approved for uveal melanoma just in August of last year. Okay. And again, this is another tool. I suspect that we're going to be seeing trials about, can we do this in the preventative setting to improve outcomes? Nothing is open yet, but there are certainly discussions. Okay, so that's basically kind of strategy one. Let's see what works in the metastatic setting and then try it in the preventative setting. Our option 2 is to perform what I call signal-seeking adjuvant trials, and then take the most promising therapies forward in confirmatory trials. And here I want to talk about Ipi/Nivo. The difference between 1 and 2 is for 2 here, it may be that we have to take therapies that we have not heavily tested in people with metastatic disease, or maybe we think the treatments will work better when there's only tiny bits of cancer in the body as opposed to kind of more bulky cancer that I can see on the scans. And so that's what kind of option 2 is here. And so at ASCO, which is the big cancer meeting just a few months ago, we reported the final results of a adjuvant trial of 2 immunotherapies, one called nivolumab and one called ipilimumab, in patients with high-risk uveal melanoma. And so this was the study design. Basically, we took people who had a high risk of the uveal melanoma coming back, and all of the patients there got these 2 immunotherapies, ipi-nivo, immunotherapies that are commonly used for cutaneous melanoma. This is the regimen that gets us the 45 to 50% cure rate in skin melanoma. And then we followed them. This was not a randomized trial, so we used what we call a real-world data set to create a synthetic control arm. So we didn't randomize patients, but we used patients who were treated previously and tried to see, did the patients on this trial do better? I would say that the historical outcomes for ipi-nivo in this disease has been assessed in a couple of small prospective trials, and we've looked at this in a couple of retrospective trials. And the likelihood of major tumor shrinkage isn't that great, maybe 15, 20% at best. The likelihood of preventing tumor growth, it's not that great, right? It only seemed to control cancer for a few months. But we hypothesized here that if we gave this treatment earlier, that maybe the outcomes would be better. And in fact, this is what we saw. If we looked at the patients treated with ipi-nivo at 3 years, 70% of the patients were doing well, no evidence of cancer. Whereas if we look at our control arm, it was 43%, right? So that was a significant difference or improvement for ipi-nivo versus that historical control. And so I think I'll kind of, and there are a number of other seeking adjuvant trials, but I just wanted to highlight that one. That's one of the more recent ones. And then the third thing, we've already heard a little bit of, or a lot about this from George, is maybe we can perform what we call signal-seeking neoadjuvant trials. And neoadjuvant means let's give a medicine before treatment of the eye and then take the most promising therapies forward. The benefit of giving a treatment, a medicine before treatment of the eye is that we can see, does it work in shrinking that eye tumor, right? Because maybe if the treatment actually helps to shrink the eye tumor, maybe those are the patients who are going to benefit the most. And maybe those are the patients who should be getting more of that treatment after the eye is treated. And so I'll talk a little bit more about the Darrow trial. So again, George spoke a lot about the mechanism, so I'm not going to kind of harp on that. I will say we've been looking at the idea of targeting PKC for a long time in ocular melanoma. So the early papers came out in 2010, 2012. And so this has been definitely of high interest. We've done trials looking at PKC inhibition by itself. Again, these were published in 2020 and 2023, but we were doing these trials a long time ago. And the likelihood of major shrinkage for PKC inhibition alone, so that one pill by itself, it was also pretty modest, like 3% with this first generation PKC inhibitor, 13% with Darrow, which was called LXS196 before. And there were some toxicity challenges. So, I was a little bit, I would have liked to see more. But as George said, IDEA's done a really great job looking to see, can we combine Darrow with other therapies, and will that lead to better outcomes, right? And so I want you to look at these plots, these waterfall plots, right? Again, the lines going down means the tumor shrank. So here, maybe a third of the lines go down. Here, maybe like a little bit more than half of the lines go down. But this is the waterfall plot when Darrow is combined with another pill called crizotinib. So we use this combination therapy where, look at this, almost every single patient has tumor shrinkage, right? And then this data set, 30% of patients have major shrinkage. That's really durable, right? And I will say that this sort of waterfall plot, I've been studying this disease for a long time. We've never seen this before. So this is really, really striking. And so what was also really cool is that in some of the early trials, some of the patients who were treated still had the eye melanoma, the primary lesion in place that was not treated. And so George showed some examples here. Here's some other ones, right? So here's a 70-year-old patient who had the primary eye melanoma there that you can see on PET scan. And just after a couple of weeks, you can see the brightness is bad. That just means it's fairly active. Here, the brightness is much less, right? Which shows that it's less active, and it also shrunk down as well. And here's another patient, 50-year-old, there's a tumor there. And after 5 months, significant shrinkage, right? And so that led Anthony Joshua to present his trial. So this is a small kind of signal-seeking trial that he did in Australia called the NATEM trial. And basically what he did was he found patients who had a tumor that would require an enucleation. Initially, he would just treat with Darrow for a month, do the surgery, and then maybe give more Darrow. And then once he showed that was feasible and safe, he found patients requiring enucleation, gave up to 6 months of Darrow, did definitive treatment, and then had the option to do more Darrow. And so this is the slide that George showed you already. You saw this, but I would just highlight that of the 12 patients who were treated on that expansion cohort, all 12 of those had a tumor that would require an enucleation. After the treatment course, only 3 required the enucleation, which means 9 patients had their eye saved, which is pretty cool, I think. And so then that's led to the trial that George spoke about. So I'm not going to spend any more time here. But we are really excited about this one. So this is just the landscape of where we are with recent and ongoing adjuvant and neoadjuvant trials. Up top are the neoadjuvant trials, including the Anthony Joshua trial in Australia, which has completed accrual, the ongoing Darrow trial, and then the neoadjuvant TEBI trial, which I didn't talk too much about, but that's going to be performed at Jefferson and should open up shortly. And then we have the preventative adjuvant trials. I spoke about the Ipi-Nivo trial. There's this vaccine trial that has completed accrual. So I'm looking forward to seeing those results. There's some more data about adjuvant Sutent, alone or in combination with another pill called valproic acid. The first cohort results was reported. There's additional data that should be reported shortly. And then we have the TEBI MRD trial, the big adjuvant European ATOM trial, and a few others as well. And so where we are overall here, strategy 1, let's take what works in metastatic disease, try it in the preventative setting. I talked about TEBI. And so the ATOM trial will evaluate the idea of 6 months of adjuvant TEBI in a randomized trial. In terms of the signal-seeking adjuvant trials, we have Ipi-Nivo, which looked really good in that single-arm trial compared to historical controls, is driving discussions to see, can we actually do a randomized adjuvant Ipi-Nivo trial, which is the only way to really tell, does our preliminary results, is it real? And then for Darrow, we have the signal-seeking neoadjuvant trial. I would love to hear George's thoughts about when we might expect a true adjuvant trial. Not to put you on the spot, George, but I think that's something that should be considered a high priority. And lastly, I wanted to talk just really briefly about the importance of real-world data. I showed you that Ipi-Nivo trial and how we use historical data sets, right, to get a sense of, are the results good or bad? And so generating that sort of data is really, really important. As I think the prior discussion, having the voice of the patient and the caregiver community is super, super important, right? And so there are a number of efforts that are ongoing to rigorously collect that information. The current site has their rare registry, as you know. So it's a patient-powered registry where patients will go online, consent, and provide their own data. The Melanoma Research Foundation CureOM group has another patient-powered registry, which is ongoing. Bill Harbour leads a group called the Collaborative Ocular Oncology Group. So that's a group of ocular oncologists and medical oncologists putting data in a database so that we can learn. And we also have this International Ocular Melanoma Natural History Study, the OMNI study, which is a global effort that I work on with Anthony Joshua and Joe Sacco, where we have nearly 1,000 patients enrolled. And I think we need other avenues to learn beyond prospective clinical trials, and this is a mechanism to do that. And using these kind of registries, I think we're going to be able to answer questions regarding, are we actually educating patients and caregivers the right way? Are we doing the shared decision-making that we should be doing? What are additional concerns that we may not be addressing, right? From the clinician registries, we can look at these various correlations that are hard to do with these single center databases. And if we ever combine these data sets, combining patient preference with clinical outcomes, I think could be really, really powerful. So I'll leave it at that. I do want to, again, just thank all of you for listening. Danae, I'd like to thank you for putting this together. I think this is really critical. We're a team, and I want to work with you to fix this problem. So I will stop
Well,
share.
Dr. Carvajal, thank you so, so much. This was so exciting to hear, and I hope that everybody who's listening is hearing and understanding your excitement just around the information that you guys are seeing, the data that we're seeing, and just the potential that we have for some of these newer treatments, and some of the ones that we're just seeing used in new ways. Let's go ahead and move on to Dr. Cole again from IDEA Biosciences is back again to discuss the daro-crizotinib combination trial and just give us a little more of an update on where the metastatic treatment of the IDE 196 is going.
Absolutely. Let me share my screen here. All right, everyone, let me know if the screen here is blocking some things. It didn't do this the last time. Again, thank you for that wonderful introduction, and my goodness, Richard, thank you very much. I hope to be able to answer some of your questions. If not, I will definitely be back in touch with you. But let's take a look at some of the things that IDEA and or darovasertib is doing in the metastatic uveal melanoma setting. Again, I'm going to give some information, which Richard already showed a little bit of in terms of our phase 1/2 clinical trial, combination of darovasertib plus crizotinib. And again, as was mentioned before, it's very important to keep in mind that what I'm about to show you in some of the later slides after we look at some of this data, this is single agent data. So phase 1, phase 2, not phase 3. So when we look at our patient characteristics in this trial, and this data was presented last year at ESMO, important thing to keep in mind in terms of our phase 2 trial here, when we look at the severity of disease in our patients. So when we look at one of our markers, LDH, 60% of our patients in the any line cohort, meaning patients who are not frontline, had a increased LDH, greater than 60% of those patients had an increased LDH, whereas our patients in the frontline cohort, 50% of those patients had an increase in LDH. If we look at our tumor sizes down here, the largest metastatic lesion, the majority of the lesions in both of our cohorts were between 3 to 8 centimeters. And also, if we look at the location of the metastases, greater than 50% of the patients in both of our arms had both liver and extrahepatic or outside the liver metastatic disease. So these patients have a pretty high severity of disease that were in our phase 1/2 trial. And they're looking at our phase 2 data right now. So again, this is a waterfall plot that we've seen before, but this one's sort of divided up into 2 different areas. And again, we want to see bars down, not bars up. And so by looking at these 2 things, we have a lot of bars down, and that's a good thing. The plus signs talk about confirmed response. In a single arm trial, we confirmed response with 2 CAT scans. So 2 cycles, these patients had 2 CAT scans confirming that response. Top waterfall plot is frontline, bottom waterfall plot, any line. So patients, this is a combination of frontline and patients who have more than 1 line of therapy. The confirmed overall response for our frontline patients, about 45%. And importantly, our disease control rate was about 90%. So disease control rate is a criteria that we use that encompasses stable disease. Another thing that I want to say, when we take a look at tumors that had greater than 30% shrinkage overall, 12 out of 20, it was 60% of the patients. But what's exciting to me is our disease control rate here in this single arm study, about 90%. Stable disease, as we've now, I would say, modern oncology drug development, modern thinking, stable disease is not a bad thing. It truly means we still have that tumor under control. When we look at our any line patients down here, again, patients that are more heavily treated, confirmed overall response rate's about 30%, but our disease control rate, again, is about the same as those frontline patients, about 90%. Again, very encouraging single line data. And again, down here, where we're looking at this, our heavily pretreated patients, 62% of these patients in this waterfall plot that we're taking a look at down here had 2 or greater prior lines of therapy. So, and I'm sorry about this on the side here. I couldn't get that to shrink, but what we're trying to show here is that even in our trial, we are HLA-A2 agnostic. So we had both HLA-2 negative patients and HLA-2 positive patients enrolled in this trial. And what's interesting for us is when we take a look at our response rates, again, single arm data, the response rate for our frontline patients about 42%. And when we take a look at our HLA-2 positive patients, frontline patients, there's a 60% response rate. Both patients have 100% disease control rate. When we look at any line patients, HLA-2 negative, we have 29% or almost about a 30% response rate with about a 94% control rate. And when we look at any line in the HLA-2 positive patients, about a 32% response rate with an 84% disease control rate. When we look at our, I'll say, molecular responses with the circulating tumor DNA, again, when we, and this is regardless of BAP1 mutation, again, another mutation which portends to more aggressive disease or poor disease or a bad tumor marker, as we like to say. We're showing pretty robust responses here. Again, single agent, single arm trial with patients, whether they are first line patients and or pre-treated patients. And again, keep in mind, this is our combination, darovasertib and crizotinib. And so one thing that we have to remember with our uveal melanoma patients, as Richard showed and as I showed a little bit before, darovasertib is really shutting down signaling through PKC. Another route of signaling with uveal melanoma is through MET. And most of our patients, another oncogene, most of our patients are showing increased signaling through MET, and crizotinib stops signaling through MET. And in Richard's slide, he showed that aspect of the MET signaling too. So what we like to think in terms of this combination, we're shutting down all signaling that is driving this tumor. So when we look at progression-free survival in these patients, and again, it's any line. When we look at the frontline patients and or any line patients, about the same amount of progression-free survival, about 7 months. Frontline 7.1 months, specifically any line 6.8 months. For patients who only have liver disease, it's about 11 months. The treatment duration for this, 50% of the patients had treatment greater than 6 months, and about 30% of the patients had treatment for greater than 1 year. When we look at our Kaplan-Meier curves, and again, we have to take this slide with a little bit of grain of salt because the darovasertib crizotinib data is not phase 3 data, but we're trying to see where this falls into place with some other phase 3 data. And I would say when we look at this Kaplan-Meier curve compared to some other curves, this gave us the confidence and the wherewithal to conduct our phase 3 trial. But we do see the PFS curve, and I think the most important thing to keep in mind from this side, if we look at that control arm where the darovasertib arm is. So again, when we look at this slide, again, we're not comparing cross, but we're just taking a look at some numbers. I would say this slide, along with the curve that I just showed you, gave us that confidence to move forward with our phase 3 trial. So when we look at our potential tolerability, safety, something that was mentioned before, when we look at our grade greater than or equal to 3 AEs that were drug related, about a third of the patients in our phase 1/2 trial. When we look at tumor shrinkage, we have a robust amount of tumor shrinkage. When we look at ORR, again, frontline, about 45% of the patients had a confirmed overall response rate. About 30% had a confirmed overall response rate. Again, this is by RECIST 1.1. And when we look at our PFS, about 7 months. So what does our phase 2/3 trial look like? This trial is currently open, currently running. And I can say we are HLA restricted to HLA-2 negative patients only. And that's because, Tevi, good therapy, is already approved for HLA-2 positive patients. So this is a schema of our phase 2/3 trial. We are currently in the phase 2 portion right now. What we're doing now, and because of some guidance or recent guidance, I would say from the FDA, the phase 2 portion of this trial is sort of divided into 2 parts, phase 2A and a phase 2B. We're having a dose optimization part of this trial at first to figure out the appropriate dose for darovasertib in combination with crizotinib. I think we have an idea of what that dose should be, but in order to make sure that we test it appropriately, we're testing 2 doses. We plan on having a meeting with our independent data review committee, and that meeting in a blinded fashion will help us determine what is the best dose. We're looking at both efficacy and safety to do that. Once we have confirmed what our dose is going to be, we're going to continue to enroll patients at that dose level to complete the phase 2 portion. The patients that were at a different dose level will then be allowed to move up to that other dose level or down as that dose level was chosen. Our goal is to have accelerated approval after the phase 2 portion. And that's going to be after that. We are simultaneously enrolling patients in the phase 3 portion, where the primary endpoint is going to be overall survival. And similar to Dr. Correa had mentioned before, we do have fast track designation for our combination with darovasertib plus crizotinib and metastatic uveal melanoma. And so this is our overall clinical development strategy. Our plan is to, yes, move forward in the neoadjuvant setting with both HLA-2 positive and HLA-2 negative patients. Yes, we do have a adjuvant plan. I can say Richard with a big smile. I'm sure I'll be able to talk with you in the next couple of weeks. But since ASCO, myself and my lead statistician have been vigorously designing a adjuvant trial. And over here, we see what we're doing right now in terms of metastatic uveal melanoma. The trial that I just presented data for and spoke about, we've just amended that clinical trial actually to enroll an arm with HLA-2 positive patients. And that ends this portion of my presentation.
We are going to bring on Dr. Vukovic from the Delcath Systems company, and they're going to talk a little about the FDA approval of Hepzato. So thank you, Dr. Vukovic, for being here.
Thank you very much, Danae, for inviting me, and happy to be here and have the opportunity to a couple of things about Hepzato and focus trial and what's next. So I'll start by sharing my screen. So again, I'm going to tell a couple of things about the Hepzato kit. And my name is Vojo Vukovic. I'm Chief Medical Officer at Delcath Systems. Dr. Carvalho has already kindly introduced, so I'm going to go relatively quickly to the first couple of slides. The Hepzato kit is basically a drug-device combination. The relatively complex schema shown here on the right shows it's really essentially a system of catheters that's inserted into the patient's blood vessels to isolate the circulation in the liver and to allow for a high-dose chemotherapy to be administered just to the liver. And then the blood that flows out of the liver is isolated and taken to an extracorporeal circuit to filters, which remove close to 90% of the chemo that was given, meaning we are achieving a regional focused high-dose chemotherapy of the liver, which of course in patients with the uveal metastatic melanoma is of extreme importance because 90% of patients with metastatic disease will have liver lesions. And actually half of metastatic uveal melanoma patients have liver lesions only. And unfortunately, most of the patients who succumb to the disease do so because of progression in the liver that cannot be controlled with systemic therapy. And that is the reason why local regional therapies, such as percutaneous hepatic perfusion, which is basically enabled with the Hepzato kit. The other component of the kit is the drug melphalan. It's an old drug. It's actually older than I by about 2 years, and that's something. But old doesn't mean bad. The reason why old drugs, chemotherapies were abandoned in favor of modern drugs, immunotherapies, is that not the lack of efficacy. The chemotherapies are very efficacious. Problem is toxicity. And toxicity means that you're not only hitting the tumor, which is the desired effect, but unfortunately, chemotherapy, when given systemically, will also hit targets such as our organs, blood vessels, and the whole body. Basically, it's making the patient sick. So if we had a means of delivering high-dose chemotherapy specifically to the tumor site without damaging too much the rest of the body, chemotherapy could really be quite effective. And that's what I'm going to show you in the next slides. So the Hepzato kit is utilized with a team of specialists under left, you see the team of specialists conducting the procedure, PHP or hepatic perfusion procedure, which is led by an interventional radiologist, the anesthesiologist, and the perfusionist. And in addition to that, of course, is the clinical treatment team led by the oncologist and other people who help prepare, treat, and manage treat the patient, and then also manage the patient between treatments with the HEPZATO kit. The 3 steps of the procedure are the isolation, followed by a local administration of the high-dose chemo, and then finally filtration to remove the vast majority of the administered chemo so that the systemic circulation receives only 10–15% of the administered dose. The product was approved by the FDA in August 23, as mentioned earlier by Dr. Carvajal. And the rationale, the basis for the approval was the pivotal focus trial, which was published, and you see the reference for the publication below. And the history of the trial, it was designed originally as a phase 3 randomized multi-center prospective trial, where patients would be randomized to receive treatment with the melphalan HDS or HEPZATO kit, or the best available care investigator choice, which included another regional therapy called TACE, pembrolizumab, belimumab, or dacarbazine. About 85 patients into the trial, we noticed a significant slowdown in recruitment because patients who were randomized to the control arm started withdrawing consent to that level that it jeopardized the randomization and the statistical design. So we met with the FDA, discussed with them the problem, and the FDA proposed that we amend the protocol to make it a one-arm trial, single-arm trial, to drop the control arm, because otherwise the integrity of the trial couldn't be preserved. So that happened with an amendment, and the study has been completed as a single-arm study, where all enrolled patients would receive the treatment with melphalan HDS, and the treatment cycles are administered typically 6 to 8 weeks apart. Inclusion criteria, basically, as far the extent of disease, both patients with liver only and extrahepatic disease as well were eligible. Insofar when 50% or less of the liver would be occupied by the tumor, tumors have to be measurable, and patients have to be in a good performance status. If the liver was compromised by cirrhosis or portal hypertension, that was a exclusion criteria. And because the procedure is conducted under general anesthesia, the patients have to be fit enough to be able to undergo general anesthesia procedure. And the patients were on immunosuppressive drugs or had infections were not eligible because melphalan would suppress the bone marrow and make patients more likely to experience infections. So the primary endpoint of the study was a response rate, confirmed, independently reviewed response rate. And a historical control requested by the FDA was a meta-analysis of contemporary trials where immunotherapy, ipilimumab, and nivolumab, or the combination thereof, was used to treat patients with metastatic disease. Based on the meta-analysis results, the response rate in this historical control was 5% approximately, and the response rate in the focus trial was 36%, which was highly statistically significant. Equally important is not just the large number of patients who responded to the treatment, but it's actually the duration of response. The median duration was 14 months, which is, depending on which trial you look at, between 3 and 5 times longer than what you typically see, except for long-term survivors on immunotherapy. Here's the complete data set. What is probably very interesting for this group, all of these are patients, both treatment naive and pre-treated patients. We saw 7 responders, patients who had complete response. And I'll show you in a graph in a moment that, and actually, why don't I go there right away? On the waterfall plot, you see that the majority of the patients respond with the tumor shrinkage. But when you look to the far right, you see that total patients had complete disappearance of the target lesions, 7 of which were classified as CRs. That means there was no tumor left in the body, and 5 were not classified as CRs, meaning there was still extrahepatic disease left over. That really says a lot about the efficacy of the treatment in the liver. But what's really also very important is on the previous slide, I'll just go back there for a moment, is the disease control rate. So close to 74% of patients achieved disease control, which is, as Dr. Cole mentioned previously, is really important. The controlling the disease, having the potential prolonged life, is really what you're trying to accomplish in this setting, in the setting of metastatic disease. So that's the efficacy data. In addition to that, I'm showing here also progression-free survival, which is 9 months, which is, I think, very, very good. I don't know of any published data that shows longer PFS in metastatic uveal melanoma. The overall survival is also very encouraging, more than 20 months. Obviously, the limitations are that this is not a randomized trial, so we cannot make definitive statements, but it's encouraging to see both progression-free survival and overall survival trending in the right direction. Here's the safety summary, and I'll show you a more detailed slide as well. In the FOCUS trial, the median number of treatment cycles was 4. We planned up to 6 treatment cycles, and about 37% of patients completed all 6 treatment cycles. We didn't record any deaths related to study treatments, which is very important because the previous method used to deliver high-dose chemotherapy to the liver, called IHP, or isolated hepatic perfusion, had a mortality rate of around 5% historically. Obviously, PHP is a less invasive, repeatable procedure, and it's better tolerated. Approximately 41% of patients had serious adverse events, which is very typical for pivotal studies in advanced metastatic disease. Typically, patients report around 40 to 50% SAEs. So this is in line with other phase 2 trials. The predominant toxicities experienced by patients with transient non-cumulative hematological toxicities. Melphalan is a drug that will suppress the bone marrow, and although we are able to filter out more than 80%, close to 90% of the administered melphalan, there's still some residual melphalan which is causing this transient non-cumulative hematological toxicity. However, hematological toxicity is something that doctors are very experienced with managing, and it responds well to standard supportive care therapy. 18% of patients had AEs that led to treatment discontinuation, but this is, again, a very strict definition. So all the patients who discontinued due to AEs were planned to receive 6 treatment cycles. So that's why the rate is a little bit higher than what you would normally expect. And what's important, although we give the treatment directly into the liver, people ask, well, is this high-dose chemotherapy toxic to the liver? And the answer is no, because less than 10% of patients experience transient grade 3-4 liver AEs, which predominantly increases in liver transaminases, enzymes that indicate potential damage to the liver. In terms of specific treatment emergent serious adverse events, and these are events related and unrelated to the treatment, you can see here on the left the table. Thrombocytopenia, neutropenia, leukopenia are the events that happened most frequently. We had 3 patients with cardiac arrest, none of them fatal, and none of them related to treatment. Some of the side effects happened during the treatment, for example, hypoxia, pulmonary edema, because you have to remember these are patients who are in general anesthesia, and hypoxia, pulmonary edema happens in a small percent of patients, and it's related to the anesthesia procedure. And then after the procedure, in the bottom right table, you see, again, the mostly hematological toxicity, as previously mentioned. So that's the FOCUS trial. And people ask us a question, okay, what's next? What's next for HEPZATO KIT in patients with uveal melanoma? And we do have plans. I think the most imminent projects that we're currently working on are combinations with immunotherapies in patients with metastatic disease. Combining chemo with immunotherapy has proven very successful in many solid tumors. So we were hoping to see a similar effect in patients with metastatic uveal melanoma, and I'll show you 1 or 2 slides how this is shaping up. And then, again, as mentioned by Dr. Carvajal earlier, we're also interested in looking at the efficacy of the treatment in patients with high-risk metastatic disease profile, meaning patients who are successfully treated for the primary tumor but unfortunately have a high risk of metastatic disease. There's lots of trials underway, and we believe that treatment with Hepzato kit has a really high potential to mitigate the risk of metastasis. And we are discussing some proposals about launching such studies, but that's something we'll probably discuss next time in a forum like this. But in the meantime, for conclusion, let me show you how the combination therapies may look like in patients with metastatic disease. Study called the Chopin trial was initiated in the Netherlands about 4 years ago as a phase 1b, phase 2 study. Currently, the phase 2 is completing enrollment, and we expect the results from the phase 2 randomized trial to be available sometime next year. But what was published about 2 years ago are results from the phase 1b, the dose finding part, because PHP or Hepzato kit was combined with ipilimumab and nivolumab, the two most effective checkpoint inhibitors thus far. And you see here the spider plot, basically how the tumor size changes following this treatment. It's very encouraging to see that the vast majority, although it's a small number of patients, it's only 7 patients, but we see definitely very encouraging signs of tumor shrinkage. And on the next slide, we see a very interesting, intriguing signal of efficacy with response rate, very high as shown here. Again, a caution, this is a small number of patients, only 7 patients, and the larger trial that is currently completing enrollment will hopefully provide a much more conclusive data set. What is also very intriguing is shown in the table on the right, and that's the progression-free survival. If you recall, if you use systemic treatments, typically you'll see 3 months progression-free survival, or maybe with some of the novel drugs like the IDEA drug, maybe 7 months. Here we have seen in this very small initial trial, 29 months. Again, this is just something to intrigue our curiosity and then really make us wait for the randomized phase 2 data, which will be available next year. So that's all the technical sets that I had here. Then I thank you very much. Thanks to the audience.
Thank you, Dr. Vukovic. If you guys have questions, please drop them in the Q&A. We'll get to them after our final presenter. I'm going to go ahead and bring on our next speaker from Replimune to talk about their data that was presented at ASCO this year. Dr. Olsen-Brown, thank you so much for being with us.
Thank you so much for the invite. I'll just share my screen. So as I said, thank you so much for the invite. I'm Anna Olsen-Brown. I'm an oncologist, medical oncologist based in the UK, and worked with Replimune on a number of their oncolytic virus studies. So I'm just going to give a little bit of background around oncolytic viruses in general, and then talk about a study that was presented at ASCO this year. Is my disclosures. So obviously throughout this entire really frankly amazing webinar, we've heard about all different types of therapeutic approaches to managing ocular and uveal melanoma. And I think one of the things that's always really important is that we understand and have a sort of moment of reflection on how quickly this field is expanding, particularly thinking about how we sort of diversify and harness the power of the immune system to try and treat in as we've heard, the neoadjuvant, adjuvant and metastatic setting. And oncolytic viruses are one of those subgroups of therapies that we are working very heavily on to try and understand where they might have a role in the management of multiple different malignancies, but that actually includes uveal melanoma. And when we think about how oncolytic viruses work, essentially they actually do a number of things. So the virus I'm going to talk about is something called the RP2 virus. But it's based on the principle that oncolytic viruses can infect malignant cells and cause direct cellular destruction, but also that leads to a series of other downstream events, which essentially leads to recruitment and efficacy of CD8 T cells, probably amongst other immune cells that we can also cause optimal recruitment via the introduction of an oncolytic virus. Generally speaking, at the moment, and certainly the virus I'm going to talk about is directly injected into an area of tumour, and it causes these localized effects. And certainly the viruses that we've been working on have been used in combination with checkpoint inhibitors in the aim to optimize that immune response. So the virus sort of allows direct effects on the tumor cells, but also T cell recruitment, with the principle that if we add a checkpoint inhibitor, we actually have an upregulated, much less inhibited T cell that arrives and is much more able to be efficacious in the tumor microenvironment. So there are multiple different downstream effects of the oncolytic virus, as well as the viral effect itself, which is quite amazing. And this has been trialled in a number of different settings so far. And this is a replimune trial looking at actually using the RP1 virus, which showed that it was very useful in the setting of PD-1 failed treated cutaneous melanoma, and we saw some really promising signs of efficacy there. And so the question was, could we put that and introduce that into uveal melanoma? So as we've talked about throughout the day, the kind of the complexities of the uveal melanoma malignancy, particularly in the metastatic setting. We know that it's a very challenging malignancy to treat, and it doesn't respond in quite the same way as its cutaneous counterpart. But also there are different immunological issues with uveal melanoma in the metastatic setting. It's quite a cold tumour. It's quite difficult to get T cells and other immune cells into the microenvironment. And also understanding whether those T cells are efficacious once they get there is also a challenge. So trying to sort of address some of those issues has led to the development of a series of oncolytic viruses, but this one is called the RP2 virus. It's a genetically modified HSV1 virus that has got a number of different mechanisms by which it can optimize its immune modulation. And that includes for the RP2 virus, this sort of anti-CTLA4 antibody-like molecule, which allows some initial checkpoint antagonism as part of the viral cascade. So the hope is that this would be seen to show some efficacy in the metastatic setting. So thinking about patients with their metastatic disease. This is going on to form part of a bigger body of work that replimune are undertaking with different oncolytic viruses, different versions of the same base, looking in various different settings. And there is a planning underway to do a bigger uveal melanoma study. But this is the sort of the data from the phase 1 trial, which was a solid tumor trial, but we were going to look specifically at the uveal melanoma cohort within that trial. And this was presented at ASCO this year by my colleague, Dr. Sacco. So if we think about the design of this study, there were 2 parts to the study. One was a monotherapy dose escalation and a dose selection part, and then that went on to a second part of the study, which divided into 2 cohorts, cohort A and cohort B. And really the difference between the 2 parts of the second part of the study was when you would introduce the checkpoint inhibitor. Did you need that up front? Or can you introduce it later on with the same outcome? And we're still answering that question. The study's still ongoing. So RP2, as I said, is an oncolytic virus. It's directly administered into the tumour, and it can be administered either to subcutaneous or superficial lesions or to deep and visceral lesions using image guidance. And that was either by a CT-guided injection into the liver, predominantly, or ultrasound-guided injection into any lesions in the liver. Patients were included if they were over 18, if they had advanced or metastatic non-neurological solid tumours, including a uveal melanoma cohort, and that they had progressed or couldn't tolerate standard therapy. They had to have at least one measurable and injectable tumour of more than or equal to 1 centimetre, and they had to have an ECOG performance status of 0 to 1. Patients were excluded if they'd had prior treatment with oncolytic viruses, if they had a history of hepatitis B, C, or HIV infection, or if they'd had any active significant hepatic infections or prior complications with HSV1. If they had active brain metastases or meningitis from secondary to malignancy, or if they've had major surgery less than 2 weeks prior to starting the study drugs. And the primary endpoint was the safety and tolerability of the virus plus or minus a checkpoint inhibitor, in this case nivolumab. That was tracked as standard, looking at AEs, and the overall response to the virus plus or minus the nivolumab. Secondary endpoints were duration of response, complete response rate, disease control rate and progression-free survival, and the 1 and 2 year overall survival. And then we, as part of the trial, we wanted to understand the biomarker elements of this. There was a large translational component to this, with multiple biopsies taken as part of the process, and there were optional biopsies as well, to try and understand what changes the treatment was affecting within the tumour microenvironment. And I'll talk a little bit more about that towards the end. So in terms of the demographics, I think that they were the RP2 monotherapy arm. There were 3 patients with uveal melanoma, and then the RP2 plus nivolumab second phase there was 14 patients. Essentially, the monotherapy to phase 2, there weren't any really notable differences. But it's really, I think the thing that's really key is the fact that most patients had prior anti-PD-1 and anti-CTLA-4 in their treatment prior to being given this treatment in combination with nivolumab in combination with the RP2. And overall, 17.6% of patients had received more or equal to 3 lines of prior therapy. So these were heavily pretreated patients, and the majority had also received this combination of PD-1 and anti-CTLA-4. If we look at the clinical activity, which is essentially where we're looking for early markers of clinical activity in this patient group, did we see any, was really the fundamental question. Interestingly, we saw an overall response rate of 29.4%, all partial responses, and a disease control rate of 58.8%. The median duration of response was 11.5 months, with a range of 2.8 to 21.2 months. And you can see that broken down here in the table. The responses were, interestingly, we found, were observed for both HLA-A2 positive and negative patients. And obviously, quite a lot of the work that we're doing in the immune space in the uveal melanoma is focused on the HLA-A2 positive group. So we wanted to see whether actually we could see activity in both of these groups, and there was in fact activity seen. And this just breaks it down in terms of visuals for you. So you can see that there were 5 patients that showed a partial response, and a further 5 that showed stable disease over time. And actually what we're really looking for is whether we see responses in the injected and the uninjected lesions. And this is a really key question for us with oncolytic virus therapy. Can we essentially show antigen presentation that leads to further T-cell recognition of other areas that will lead to response in the non-injected lesions? And you can see in this patient here who had a partial response, you can see responses in both the uninjected and the injected lesions, which is important to notice that we do see this sort of more sort of abscopal effect, and again you can see that as well. So when we're thinking about the clinical responses, we also want to see what's happening at a more baseline translational level, because that will help us understand how we can then potentially coordinate and build on these results. So what we did here is we took biopsies at baseline screening and at day 43, and we stained for CD8, which is a marker of T-cell presence, and PD-L1, a marker of immune activity. And you can see that these change over time, that the tumours become more infiltrated in response to the injections, and also there's a higher expression of PD-1, which would suggest that there is a more sort of immune environment. Interestingly, we also looked at T-cell receptor sequencing within PBMCs within the peripheral blood, looking to see if we could see a response in the sort of the circulating immune cell repertoire as a response to treatment. And we can see that there are significant expansion and new clones. There was some clones that got smaller, but actually there are, as you can see, a lot of blue blobs on these curves, which showed that numerous clones exhibited expansion that were first identified on day 43, so after the implementation of treatment. So we can see that this therapeutic combination is leading to immune activation and actually seeing benefit. One of the things that was interesting is we didn't necessarily see that this clonal expansion correlates to clinical response. So that means that there's probably more work that we can do, and probably with additional agents or different trial design to actually understand how that will lead itself to leading to clinical response in all. But actually just seeing the fact that we see those changes means that these therapies are immunogenically active, which is really reassuring at this stage in the phase 1 trial. The safety profile is also quite interesting. So as you can see here, there were some patients who got toxicity. The most common were grade 1 or grade 2 in the groups were pyrexia, chills, fatigue, and hypertension, and some people also got itch with pruritus. There were 2 cases of grade 3 hypertension, but both were transient and regularly managed with fluid therapy alone. And there were no grade 4 or 5 AEs, which again is quite relevant. Interestingly, given the fact that the majority of the patients in the RP2 plus nivolumab trial also didn't experience any particular immune-related adverse event toxicities, is really quite noticeable and suggests lower rates than we would do with nivolumab therapy as a single agent, which is quite interesting at this point. So in conclusion, we see that RP2 monotherapy and RP2 plus nivolumab therapy resulted in an overall response rate of 29.4% and a disease control rate of 58.8% in a cohort of patients with metastatic uveal melanoma who had been heavily pretreated, and they showed durable responses and a favourable safety profile. We saw this activity regardless of HLA status, which I think is very reassuring for that patient group that don't have a HLA-2 positivity. And it doesn't seem to be specific to a prior ICI regime. Essentially, patients could have had already been treated with an anti-PD-1, and we would still see benefit in this setting. And we saw responses in both the hepatic and extrahepatic metastases and injected and non-injected lesions. So we're seeing quite a lot of activity across the metastatic repertoire even in those patients who've had only a small number of lesions injected. We do see increased immune infiltration, and we see increased PD-L1 expression in tumours. And we see also there's changes in the peripheral T cell repertoire, which is useful in understanding the fact that we see different T cells being expressed and expanded, but also we have a peripheral marker of potential immune activation, which is useful in patients generally, and might possibly have some clinical translatability further down the line. This suggests that this intratumoral oncolytic immunotherapy, with this expression of this anti-CTLA-4 molecule as part of its genetic modification in combination with this checkpoint inhibitor, has the potential to provide clinically meaningful benefits and a favourable toxicity profile. And therefore, we're going to Replimune are taking these forward and developing a more advanced phase 2 trial. So essentially, we see really notable objective response rates and disease control rates, and patients don't appear to exhibit particularly concerning safety profiles. And obviously, Replimune will be more than happy to take any requests for further information, and their emails at the bottom of this.